Smith and went on to get her PhD in chemistry under the mentorship of Bradley Pentelut, where her thesis focused on design of nuclear targeting peptides for macromolecular delivery using machine learning. She has been prolific along the way kind of already just a short career with 15 publications and being the author of 4 patent applications. She's a recipient of prestigious Miller Institute Research Fellowship and is doing and in doing so had to turn down actually an F-32. So anytime you're turning down money, you're doing really well. So I'd like to congratulate you on that. She's won numerous awards along the way, including MIT Department of Chemistry Award for Outstanding Teaching, numerous Best Talks and Poster Awards, and the UC Berkeley Postdoc Achievement Award. So congratulations on that. Highlighting just 2 research achievements, her work has led to the development of mass spectrometry-based profiling platform as a new method for characterizing uptake and cellular localization of D-peptide antisense oligomer conjugates, and the discovery of the first chemical reaction to install a carbon-carbon bond within the peptide backbone. With that, we can start your talk and be talking about ribosomal synthesis of diketone-containing peptide backbone via post-translational acyl shift. Take it away, Carly.
Thank you so much for that kind introduction, and again for the opportunity to come here today and tell you all a bit about what I've been doing during my postdoc. So we just heard an excellent talk about how genetic code expansion can reveal biological function. And now I'm going to flip the script a bit to talk about how we can use chemistry to expand GCE beyond your typical alpha amino acids. So if we're talking about things that are not alpha amino acids, there's actually been very few examples of these monomers being incorporated by cells. And so what this might look like is, in cells or outside of cells, ribosomal generation of new bonds. And some monomers that have been incorporated along these lines include alpha-hydroxy acids, beta amino acids, beta-hydroxy acids, as well as a recent di-substituted alpha amino acid. So these are all extremely interesting, and the way that these monomers are incorporated is the same as with alpha amino acids. So here's a cartoon of the ribosome, and zooming in on the PTC, we can see that the incoming amino acid has a nucleophile here, X, which attacks the ester of the growing peptide chain. And so in nature and in a lot of the genetic code expansions that we use, that X nucleophile is an amine. And so the product of that is an amide. It's also been known for about 50 years that the ribosome can incorporate alpha-hydroxy acids to form ester bonds in the backbone. However, something that has not been shown yet and is thought to be impossible is incorporation of carbon nucleophiles in the PTC to generate ketone backbones. And so right here, this would be a carbon-carbon bond forming a ketone, and this motif would impart some really interesting structure and reactivity embedded within the peptide backbone rather than just on the side chain. So not talking about the ribosome anymore, but still just incorporating ketones into peptides and proteins is pretty challenging. But there's 2 key ways that people do it so far. Jorn Peel's lab published on this really interesting tailoring enzyme, which can excise a tyramine residue and leave behind an α-diketoamide. And so right here, this is a ketone that can then be labeled using bioconjugation. Some drawbacks here, though, include that this enzyme requires an 11 amino acid recognition tag. So if you're interested in doing this on smaller proteins or peptides, that would be a major limitation. And then you also lose the neighboring stereochemistry of this amino acid. Thinking about synthetic approaches, you can couple ketones directly onto peptides and proteins. But typically, this is coupling done on the side chains or termini only, and it's not site selective. This would be typically on any amino group that you have available. Okay, so thinking about chemistry kind of beyond classic coupling, acyl shifts are actually really powerful for backbone modifications, and you all are already very familiar with this strategy. So inteins in nature, as well as chemists using native chemical ligation, take advantage of O or S to N acyl shifts. So what this might look like is the side chain serine or cysteine forms an oxo or thioester linkage with some other, a separate peptide or protein. And then the free alpha amino group then attacks that ester and rearranges and reinstates this canonical alpha amide backbone. Okay, so this is used for protein splicing via an O to N or S to N acyl shift. Our group recently published on a similar strategy where we actually switched the atoms here. So if you start with an alpha-hydroxy acid and you have a beta amino group, when you unveil this beta amine, a similar O to N acyl shift occurs, but it actually installs a beta amide linkage. And so this is a way to install a beta amino acid without actually needing the beta amino group to be the nucleophile in the PTC, because we know that installation of beta amino acids can be really challenging. Okay, and the reason why these reactions are so efficient is because they take advantage of a 5-membered ring cyclic intermediate. And so my question here when approaching this project is, can we use a similar acyl shift strategy to actually install a carbon-carbon bond in the backbone of peptides? And so really this means if it's too challenging or we haven't been able to yet to incorporate carbon-carbon bonds directly in the ribosome, can we actually install them post-translation? And so the inspiration for this project came from dehydroalanine, which is an alpha amino acid, but it's sort of unnatural in that it has this alpha beta unsaturated amide group here. And so this acts as a Michael acceptor and actually allows you to install really unusual side chains there. And so inspiration from that resulted in dehydrolactic acid, where instead of this amide linkage here, now we have an ester. And so my question really was, would this double bond still be a Michael acceptor? Because now with this being an ester, this motif is called an enol ester, and it's actually quite reactive. And so there's a variety of reactivities that we could imagine here, but I'll just give you my main question was, is the chemistry of DHL resemble that of DHA? And so this is a synthetic slide for how I make this monomer, but I start with this glycidate, and I use diphenyl diselenide to do an epoxide ring opening step. And then I cleave off this methyl ester to give this alpha-hydroxy acid, which I call alpha-hydroxy phenylselenocysteine. And using phenylselenocysteine is a really common approach for making dehydroalanine. And then this hydroxy acid can be incorporated into a model tripeptide, where I first protect the C-terminus with an amide group, and then I can couple on a variety of Bach-protected amino acids on the N-terminus. And just like with dehydroalanine, I do a hydrogen peroxide treatment to eliminate the phenylselenide side chain, leaving behind this double bond, and now this is a DHL model peptide. So this model peptide is stable in organic and acidic solvents. So I was able to purify it by PrEP HPLC, and it gives this single peak here where the black line is the total ion chromatogram from LCMS, and the gray peak is the extracted ion chromatogram corresponding to the mass of the DHL peptide. Okay, and then something really crazy happened when I added base to this molecule. So when I put this molecule in sodium phosphate buffer at pH 7, I actually see rapid isomerization of those peaks. And so the extracted ion chromatogram here is the same, where I'm extracting for the same mass, but now you can see that molecule has changed. So I wanted to look more into what was actually happening in this isomerization, and for that I used NMR. Specifically, heteronuclear multiple bond correlation NMR really revealed the through space interactions and couplings of protons and carbons. And so here I'll just highlight two signals that I see. So this one in blue corresponds to these benzyl protons coupling with this amide carbonyl. And then in red are these alanine protons coupling with this ester carbonyl. And so with the y-axis here being the carbon spectrum, you can see that both of these couplings are happening with carbons in the ester amide range. Okay, however, when I add sodium phosphate buffer at pH 7, we can see that the blue signal's roughly the same, but the red signal has really shifted downfield in the carbon spectrum. And so now it's entering what would be considered the ketone range. I confirmed the structure by NMR and found that it had actually rearranged into this structure. And so now this red signal is corresponds to these alanine protons coupling with a ketone carbonyl. And this is actually a diketone species or a ketoenol species. So this rearrangement was actually quite surprising, and then I wanted to derivatize it in order to really confirm its structure. So again, this structure is a single structure by NMR, but I found that it was not acid stable, so I wasn't able to isolate it, and that explains why I see so many different peaks in this chromatogram here. However, just adding hydrazine at slightly acidic pH, I see those multiple peaks coalesce into a single sharp peak, corresponding to this pyrazole structure. And so what's happened there is hydrazines come in and formed a heterocycle with these atoms right here. And now pyrazole is very stable, and I was able to isolate that and confirm its structure by NMR. And so we've been able to install pyrazoles in the backbones of peptides, and this is really interesting because pyrazoles are medicinally relevant core motifs, although they're actually very rare in nature because it's hard to form nitrogen-nitrogen bonds in nature. However, they're very common in medicinal chemistry, and here's just a few examples of drugs where pyrazoles making up their core motif, but this includes analgesics, NSAIDs, and the CDK inhibitor. Okay, going back a bit, this rearrangement is quite unprecedented, and we had to do a lot of thinking in order to figure out how this is actually forming. And so I'm going to show you some computation that really lets us think about a potential mechanism. And this was done in collaboration with Professor Matt Francis, who's also at UC Berkeley, as well as my former mentee, Hannah Kong, who really wanted to learn a bit more about computational chemistry. And so these calculations were performed by them. So we start with this model DHL peptide, and either this molecule acts or it tautomerizes into this species here. But either way, the nitrogen atom of the amide we think is acting as a nucleophile and attacking this ester carbonyl here, which forms this first cyclic intermediate. And this first step would not be surprising to us at all because this happens in nature as part of protein degradation in isoaspartate formation. So this type of cyclization and then ring opening can form isoaspartate. Okay, and then when this ester bond breaks open, it actually reveals something that looks like an enol. And so you might be familiar with this when thinking about Claisen condensation or PKS formation in nature. And so this molecule now can either tautomerize into an unproductive intermediate, or this carbon is 5 atoms away from this imide carbonyl, which is reactive. And so this could be the crucial carbon-carbon bond forming step here. And so that likely produces the second cyclic intermediate. And then when this imide bond breaks open here, that then reveals the diketone motif. And this molecule tautomerizes into its low energy state of this ketoenol species that I showed you earlier. And so we think this two-step acyl shift reaction is the mechanism for the rearrangement that we see. So I then wanted to look at the substrate scope of this reaction, specifically looking at the effects of the N-terminal amino acid. And so these are the side chains that I looked at with varying size and substitution. And the reactions I looked at are, again, the pyrazole forming reaction. And then I also looked at labeling of ketones with hydroxylamines to form oximes. So here I'm showing you again LCMS traces where the black line is the total iron chromatogram. And then the pink peak corresponds to the product and gray corresponds to the starting material. And so we can see that all substrates give near quantitative conversion to the pyrazole, and especially those substrates which have smaller N-terminal side chains, so alanine, glycine, and then this is still a singly substituted glutamate. These show full conversion, so no more starting material observed. We can also look at this reaction with hydroxylamine labeling. So again, red is oxime and gray is remaining starting material. And while we do see labeling, you can see that there's quite a number of peaks here, and that's due to the limited stereo control we have of this oxime, whether it's an E or Z conformation, and then on which ketone it's actually labeling. But it does still achieve that labeling. Okay, I then wanted to look at this reaction in slightly longer peptides, and to do that, I needed a way to incorporate this monomer using solid phase peptide synthesis. And to do that, I made something called an ester block. And so I start with the hydroxy acid here. I protect the C-terminus, and then I can couple on a variety of FMOC-protected amino acids and remove that acid protecting group. And it gives us something that looks just like an FMOC-protected amino acid, but it's actually a di-depsipeptide because it has this ester linkage here. And so this block can be incorporated with solid phase peptide chemistry. And I generated these numbers of selenopeptides. So these peptides could then be oxidized to form the DHL analogs. And I actually found that an aprotic solvent really accelerates this elimination. And so looking here first, this is DHL formation in the presence of water. And red is the DHL peptide and gray is the starting material. And we can see that that reaction is not done after 1 hour. But when that reaction is performed in acetonitrile here, we can see that no more starting material remains, and our major peak is the DHL peptide. Okay, and so I then looked at the isomerization of these peptides and found that, as expected, isomerization is pH dependent. And so here's a DHL peptide. I incubate it in base at pH 7, 8, or 9 for some amount of time, and then add hydrazine for 1 hour in order to form this pyrazole. So here I'm showing the final time points for pH 7, 8, and 9. Again, the black trace is the TIC. Red corresponds to the pyrazole, and gray corresponds to the starting material. We can see that at pH 7, we still see a significant amount of starting material, as well as a few other peaks here that actually correspond to hydrolysis. However, when we increase the pH, we see that reaction is a bit cleaner, and then now pH 9 after 75 minutes, we predominantly see the pyrazole and very little, if any, DHL remains. And so my hypothesis here is that higher pH enhances isomerization and allows that reaction to outcompete hydrolysis, which is a bimolecular reaction. Okay, and so knowing that we can isomerize and label a number of these SPPS peptides, I tested a variety of labels. So labels that worked well and that looked interesting, for example, is this fluorobenzyl hydrazine group here, as well as phenylhydrazine and this hydrazinobenzoic acid with an additional group possibly to be labeled in this acid. Another label I found really interesting was this thalidomide hydrazine. As you might know, thalidomide is a potent cereblon E3 ligase binder. And so with this label, you could perhaps think about generating degraders with peptides. And then, as I mentioned, hydroxylamines can be used to generate oxime peptides. And then, of course, the hydrazine by itself generally forms near quantitative conversion to the pyrazole peptide. Okay, but I didn't forget our initial goal, which is genetic code expansion. And so going back to the question is, can this ketone motif actually be genetically encoded? So remember I did say, still no one has been able to form a carbon-carbon bond within the ribosome, but can we do this DHL rearrangement in order to genetically encode a peptide that can become a ketone? And so to do this, we used in vitro translation, which many of you are familiar with. But basically, this includes generating synthetic tRNA, which can then be acylated using either an enzyme or chemical acylation with the hydroxy acid monomer. And then this is put into an in vitro translation mixture where the ribosome actually incorporates this monomer into a peptide. And then for our purposes, I use a molecular weight cutoff filter and desalted to give just peptide products. And then I can do the DHL chemistry to make pyrazole peptides, which are then analyzed by LCMS. And so a lot of this work was done by Helena, a grad student who is about to pass her quals, which is very exciting, as well as Isaac Garcia, my current undergraduate mentee, who was the first to demonstrate that this monomer can be incorporated by the ribosome. And so what they did was they found that FRS1 it's an analog of pyrrolysine synthetase, which is better accommodating for phenyl side chains. And they found that it can acylate this hydroxy acid onto tRNA, and we get a mixture of mono and diacyl. These acylated tRNA products can be analyzed by fully intact tRNA mass spec, showing about a 1:1:1 ratio. That mixture of acyl tRNAs can be added to an in vitro translation mixture, coding for this peptide shown here. And we do see the selenopeptide being made by the ribosome. And so something really interesting I think about for these selenopeptides is there's selenium in them because their mass spec gives this really interesting isotopic distribution. Okay, but when we treat with hydrogen peroxide and that side chain gets oxidized and eliminated to form DHL, we no longer see that isotopic distribution. And still high resolution mass spec shows us that we indeed acquired this DHL peptide. That DHL peptide can then be labeled with a variety of hydrazines in order to form the pyrazole peptides. And so with that, this shows that we can generate pyrazole embedded backbone peptides using the genetic code and using the ribosome. Okay, and so just to summarize again, today I've shared with you a novel DHL rearrangement chemistry, which takes advantage of a two-step acyl shift to first form a new carbon-carbon bond in a diketone molecule, which can be derivatized into heterocycles embedded in the backbone. Using this chemistry, I made a variety of pyrazole-embedded peptides, and we also showed that the ribosome can make these peptides as well. And so with that, again, thank you so much for the opportunity to come and tell you all about what we've been doing. And special thanks to Alana Shepard, who's been an amazing advisor, as well as the folks at Berkeley and the entire Shepard's lab, as well as our co-authors. This work was funded by the NSF Center for Genetically Coded Materials, as well as the Miller Institute. And with that, thank you, and I'm happy to take any questions.
That was great. Thank you very much. I'm not a chemist, so I'm not of any use to ask any real questions about the majority of your talk. But now that you can encode these backbones, do you have the next step use for these in like application?
Yeah, there's a variety of applications that you can think of, but the one that I would be very excited about and that we are currently pursuing is looking at these heterocycles in macrocyclic peptides and how that might affect their structure and functions such as permeability. So that's the route that I'm the most interested in.
That's great. So you can use them as like lariats to get things into cells and whatnot?
Yeah, exactly.
Yeah, great. Linda, do you have a question?
I do. A couple of the bacteria that you mentioned, I think Streptomyces and Pseudomonas have these N compounds. And I'm just wondering, do you have any idea what they're doing?
A lot of the pyrazole-containing natural products are antifungals or antibiotics.
Okay.
Yeah, I think that those are the only examples that I found.
Are there any other questions from the audience?
I can ask a real quick one. Also not a chemist, so not qualified to ask too many questions here. But you mentioned real quick, glossed over real fast, that you were doing mass spec with whole tRNAs, amino acylated tRNAs. My understanding is that's not trivial to do. Is that true? What went into being able to develop that methodology to assay that reaction?
Yeah, I'm glad you brought that up because it's really not trivial at all. And I think very soon we'll actually have a Nature Protocols paper out really detailing that. But what it involves is a dedicated high resolution LCMS that's run in negative mode. and with very specific buffers that can chelate all of the salts that otherwise might generate a wide range of mass adducts onto the tRNA.
Interesting. Very cool.
But it functions basically similar to a peptide or protein LCMS.
Okay, cool.
Yeah, there was a lot of like gems in this. So like FRS1, how did you come across that?
So that did not come from our lab. That was originally developed to be able to incorporate unnatural side chains that look a bit more like phenylalanine rather than the pure lysine.
I got it.
And it just so happened that that synthetase worked well for our hydroxy acid after screening several analogs.